Hi everyone, my name is Morgan Ellis, and today I'll be talking to you about how to use a qPCR to detect marine invasive species. So a qPCR is fairly similar to a traditional PCR. However, we're able to use species-specific primers and probes to detect small quantities of marine invasive species DNA. As you can see from the diagram here, it follows a pretty similar method. We take our environmental sample, we extract the DNA from there, run it as a PCR amplification, and we'll get these curves, which I'll talk about shortly, and then we can process that data. qPCRs are super sensitive, and they're able to detect often less than 10 copies per microliter of DNA. And there's two main methods. One's to use a fluorescent dye, such as cyber green. This will amplify, or this will fluoresce with any sort of amplification. Or we can use a fluorescent probe, such as a PacMAN probe, which is what I'll be talking about today. It's just a little bit more species specific and gives us a little bit more confidence in the results. Is on the 5' end, it'll have this reporter or a fluorescent probe, and on the 3' end, it'll have this quencher. The fluorescent probes, there's lots of different types you can get. The most common one is FAM, which is the green fluorescent probe. You can also get HEX, which is red, Sci-5 yellow. There's a lot of different sort of types out there, so it depends on sort of the assays that you're running for which one works best for you. So the fluorescent probe works by being bound to the probe, which is a small stretch of DNA similar to a primer, which binds to our template strand during the PCR. The quencher at the 3' end, when it's in close proximity with the fluorescent probe, stops it from fluorescing. However, during the annealing stage, the DNA polymerase will come along and separate these two. And as you can see, once it floats off, it starts to fluoresce. So the more cycles this goes through, the more fluorescent probes get knocked off, the more our sample will start to fluoresce. The qPCR is then able to measure this fluorescence. As you can see here, it will come up as these curves. Once the fluorescence reaches a threshold, it'll give us the number of cycles that threshold is reached at. This is known as our CT value. So the higher the quantity of DNA, the lower the CT value will be. So you can see down here, these samples are coming up at around 25. That's a lot lower quantity of DNA than these ones, which are coming up at around 10. If we want to be able to quantify the amount of DNA in there, we can use these things called standards. Standards also act as a nice positive control. So these are a set of known DNA concentrations. We're able to plot the CT value of the standard over here on what's called a standard curve. And then this little X represents our unknown sample. We can plot that against the standard curve and quantify the amount of DNA from our known standards. This also allows us to determine a limit of quantification, which is the lowest limit that we can quantify the DNA to, and more important from a biosecurity perspective, the limit of detection. So any samples that come up below our limit of detection, we then start to doubt whether the DNA is actually present in these samples or not. From a known quantity of DNA. So that's just as simple as taking, in this example, 2 microliters from our first known concentration of DNA into 18 microliters of PCR water and doing that several times from each tube until we get to our lowest concentration. How do we know if a qPCR run was successful? The first thing we can do is look at our standard curve. We want the R² value, which is how well our standards fit against the curve, to be greater than 0.99. Our example here has it sitting at 1, which is as good as you can get, which means our standards are in a nice straight line that fit along this curve. Next thing we can look at is our efficiency. You want the efficiency to be sitting between 0.9 and 1.1. Sometimes it's given as a percentage, so between 90 and 110%. Our efficiency is down here at 96. So again, this is an example of quite a nice run. And our sample is sitting nicely in the middle of our standard curve, so this will be above our limit of detection. The next thing we want to check is samples. So we'll generally run samples in triplicate. If you have two or more positive detections, then we can have pretty good confidence that this is a positive result. And again, I just want to stress that this is a positive result that there is DNA belonging to an invasive species in the water. It's not a positive result that there is an invasive species in the water. Further checks are needed to be done, but the qPCR is a good way of being able to prioritize sites. If only one of our triplicates comes up, this is generally referred to as an inconclusive result, and some more work needs to be done to see whether we do quantify that as positive or whether we need to take more samples from the site and run them again. And if none come up, then this will be a negative result. We're pretty sure that there's no invasive species DNA in the water. And finally, we want to check our non-template control or our blank control. These are the lines down here, which are nice and flat. There should be no sign of amplification. If we do get small amplification starting to come up, this is a sign that there's been some cross-contamination. This is an example of a qPCR recipe. So this is for a 10 microliter reaction. You'll have about 5 microliters of your master mix, 0.5 of a 10 micromolar primer, and you want about half of the concentration of the primer for the probe, a little bit of PCR water, and then 2 microliters of either our sample, our standard, or for running a NTC, then just blank water. And you can see from the qPCR conditions, they're pretty similar to a traditional PCR. The biggest difference is there's generally no extension step. qPCRs have quite a short sequence, so we don't need that extra extension step to be able to finish amplifying the DNA. And the fluorescence will be read after this annealing step. I'd like to thank you all for listening and wish you luck in detecting some invasive species on a qPCR. Thank you.